we shall define what is immunogenicity. Immunogenicity is the ability of an antigen to induce immune response in the body, both humoral and or cell mediated. It means to tell that in last class when I explained to you all, I told you that immunogenicity is a property of an immunogen, that is, it has to, whenever it is in contact with the body, okay, what is an immunogen? I told it is an antigen, okay. When it is in contact with the body, if it should have an ability to evoke an immune response. What is an immune response in our body? So, the immune response is, one second, I will just enlarge the video. Okay. So, the immune response means it is the two type of cells which are involved. Okay, one is the T cell mediated immune response, the other one is the humoral immunity. For example, for humoral immunity we have the B cells, and for cell mediated immunity we have the T cells. So, when the B cells, that is B cells and the T cells which are produced from our body, when they react with an antigen, and B cell will produce an effector B cell or the plasma cell and the memory cells, whereas our T cells will produce the effector T cells and the memory cells. So, substances that satisfy this property of immunogenicity, they are called as immunogen rather than antigen. Why? Because in our earlier class I told that all antigens cannot be immunogen. We gave an example of hapten, which has a very small molecular weight molecule. For a compound or a chemical or anything or a microbial agent, if it has to be an immunogen, then it should have certain properties, and one such property is that its molecular weight should be more than 10,000 daltons. So, this condition is not satisfied by all the antigens. So, hence we told that all antigens are not an immunogens. But you know that once the antigen, okay, the property of an antigen is, though it cannot evoke, or the hapten, it cannot evoke the immune system, it can go and bind to the products of the immune response. So, what are the products of immune response? The products of immune response are this B cells, okay, and the T cells. So, that is what we have defined in the next slide. The ability of an antigen to combine specifically with the final products of the humoral or the T cell mediated immunity, it is called as the antigen, or the phenomena is called as the antigenicity. All molecules having immunogenicity property also show antigenicity, but reverse is not the true. Why? Because we saw that taking the example happens which are antigenic but not immunogenic, that is, they cannot evoke an immune response. That is the example we gave it as penicillin. So, coming to epitope, as I told you that this comes for 2 marks question, that is, they will ask you what is an epitope or what is an antigenic determinant. So, it is one and the same. So, when we told if this is a microorganism, our immune system will not recognize the entire microorganism. but only the part or the portion of the microorganism, and that is known as, we told as antigens. And what are these antigens? We told it is a protein or a glycoprotein or a polysaccharides or a nucleic acid. But here also there is specific region on the antigen wherein our immune system can go and bind, and that smallest region, okay, the smallest region or the smallest portion, it is known as an epitope. So, epitope means it is the smallest unit of antigenicity. So, how it is defined? It is a small area present on the antigen compressing of few amino acids or monosaccharides residue that is capable of sensitizing the T and B cells and reacting with the specific site of the T cell receptors or the antibody. So, this is the definition of the epitope. You can just take a screenshot of this. So, this will serve as your 2-mark answer. So, now if an antigen is having an epitope, even our antibodies, okay, they have to go and bind to this epitope. So, if this is, for example, imagine this is the epitope. I will just change the color otherwise. Color. Okay. Suppose say this is your epitope. Now, your antibody, if it has to bind to the epitope, suppose say this is the antibody, okay, this is the antibody, okay, our antibody has gone and bound to the epitope. Okay, the region, this particular region, okay, this particular region which goes and binds to the epitope is known as the paratope. So, epitope is a part or the portion of the antigen, whereas paratope is the part or the portion of an antibody. So, paratope and epitope, remember this, they will be asking for the examination. We will define this, what is paratope is, specific site of an antibody that reacts with the corresponding epitope of an antigen is called as a paratope. So, now we have different types of epitopes. One, it is called as a sequential or linear epitope. That is, they are present as a single linear sequence of amino acids. Now, what happens is, suppose say this is your epitope, okay. So, this epitope is a protein. So, you will be having certain amino acids, okay, this is all amino acids which are present. So, a linear or a sequential epitope will appear like this, okay. So, you have a linear sequence of amino acids which are present. So, you have an amino acid here, you will be having amino acids here, you will be having amino acids, but they are all in the linear form. This is called as a sequential or linear epitope. Then we have conformational or non-sequential epitope. So, these are found on the flexible region of complex antigen having tertiary structure, and they are formed by bringing together the surface residues from different sides of the peptide chain during its folding into the tertiary structure. That is, here conformational or non-sequential means they are not in a sequence, okay. Now, sequence means how, see 1, 2, 3, 4, this is all in sequence, okay. That is the meaning of sequential linear epitope. Instead of number, you just have to imagine these are the amino acids, maybe valine, glutamine, etc. like that. Now, conformational non-sequential means, see, your antigen are all three-dimensional structure. Okay. So, they be having tertiary structure. You must have read about primary, secondary and tertiary structure. So, that is whenever they are in three-dimensional structure, they are known as tertiary structure. So, now what happens here in conformational or non-sequential type, you do not find this pattern 1, 2, 3, 4, 5, 6 in sequential, but here what happens, it will be like this, okay, something like briefly I am drawing. Okay. So, just consider the sequence 1, 2, 3, 4. So, when it comes like that, just I will put here, this is 1, okay, this is 2, this is 3, okay, and this is 4, maybe this is 5. Now, girls, just compare this type 1, I mean the linear and conformational. You see that the distance between 1 and 5 is quite, okay. After 1 you have 2, 3, 4 amino acids and then it is the 5th. But when it comes to such type of tertiary structure, you can see that 1, 2, 3 we have written here, 4 it is. This is just an example I have given you how one pattern appears, and you can say that 1 is close to 2, at the same time you say that 1 is close to 3 also. Okay. So, here what is happening, now all the flexible regions, okay, that is these amino acids which are present on the sockets of the antigens, they can all come closer to each other. In which type? In conformational type. Okay. So, that is the meaning of what is sequential and what is conformational. Then we have T cells recognizes sequential epitopes while the B cell bind to the conformational epitopes. So, now what happens when our body is facing these antigens, if it is a first type, sequential type, who is going to find out that it is the T cell recognizes the sequential type, okay, that is the first linear type, whereas our B cell will find out the conformational type or non-sequential type of epitope. So, coming to the haptens, we have defined it as low molecular weight molecules that lack immunogenicity, that is they cannot induce an immune response, but retain the antigenicity or immunologically reactivity, that is they can bind to the specific antibody or T cell receptor. So, if you remember my previous slide, we had written that an immunogen can, what did I tell? It can evoke an immune response, and what is the product of the immune response? It is the B cell or the humoral immunity, okay, that is your antibody and your T cells. Okay, this is what is your immunogen is. What when we spoke about antigen is, this step is not there. Okay, but what we told that it can go and bind to the antigen or the T cell, that is what is an hapten is. Okay, it can go and bind, it can go and bind to the products without this step. But if you want to make this hapten as an antigen, then what we told that this hapten, if I want to make it an immunogen, then I have to combine it with the carrier protein. Okay. Haptens can become immunogenic when they are combined with larger protein molecules called as carrier. The hapten carrier complex is capable of inducing the immune response in the body. Hope it is clear. Just read the slide once again. these antigen are of different types. There are like self or auto antigen and non-self or foreign antigen. What is the self antigen or auto antigen means? Our own cells and molecules are called as auto antigens. For example, the lens of the eye or the sperm which is present in the male organ, these are all antigens. For example, actually our body, I told you, it has an ability to recognize which is self and non-self. So what happens, our body's immune response will not react against our own antigens. It is not going to react against our own antigen. But sometimes what happens, okay, sometimes what happens, you can look at the slide, self or autoantigens means they belong to the host itself, they are not immunogenic. Hosts do not react their own antigens by exhibiting a mechanism called as immunological tolerance. That is, during the birth itself, our body would have studied, our immune system would have studied our body. They will recognize which is our cells and which is the foreign cells, and they would have thought themselves that I should not attack my own cell. So that is the called as immunological tolerance. But what happens sometimes is the self antigens are biologically altered and can also become immunogenic. That means, suppose say if a cell has become a cancerous cell, then whatever definition we have told will not be right, because what happens, cancer cells are no more normal cells. Okay. So for another example, when we study the last chapter, you will come to know that there are so many diseases, you can see that, wherein our own immune system is going on attacking our own body, okay, and leading to immunological disorder. For example, rheumatoid arthritis. Okay. So in rheumatoid arthritis is an immuno disorder. Here what happens is, your immune system, okay, it's going to attack your own tissue. And then what will happen when it's going on attacking your own tissue? You will see that the patient will be having severe body pain, particularly the joint pains. Okay. So this is what are called as autoantigens. Non-self or foreign antigens. So this non-self or foreign antigens means mostly these antigens are they are not present in our body, they are completely foreign to us. These are all like heteroantigens. Okay, they are not a part of the human body. They can be any microbial agents or non-microbial agents also, like plants, animals, otherwise any other human product. Okay, these are all called as non-self or foreign antigens. So what is a foreign antigens means they do not belong to our body. Then what are the examples you can give is, I can give the example of microbial agents, I can give the example of plant. Why? Because we know that we were telling that sometimes when people eat certain type of plant, they become allergic to it, some food, okay, and the pollen grains also, when you inhale, you are allergic to it. The animals, okay, so some of you may be having pet animals, but you go near the pet animals, sometimes you see that you are having difficulty in breathing, okay, maybe the fur of those animals are acting as an antigens and they are disturbing your immune system. Or even other humans also. Now suppose say the excreta of some human or the animal, the fecal matter, when it comes contact with your body, that time also it can lead to a certain problem. So these are all called as non-self or foreign antigen. These foreign antigens are based on the based on the phylogenetic distance to the host. There are of 3 different types. Okay, these are the 3. Alloantigens are species specific. Tissues of all individuals in a species contain specific antigens. That is, for example, you and I belong to the same species. Okay, we are humans. So what happens, we are almost similar, but that doesn't mean that we are 100% same. There is some amount of difference, as I told you, like even humans, okay, even they can act as an not the, what I mean is by human is any some excreta or the saliva of any other human can be an antigen to us, though we all belong to the same species. Then what about isoantigens? See, one example of alloantigen I can tell you, it is the MHC molecule. No, MHC, it is isoantigens. Okay, sorry. So what are isoantigens means? Isoantigens are type of antigens which are present only in subset of species. Example, the blood group antigen and histocompatibility antigens. The histocompatible antigens are highly specific as they are unique to every individual of the species. That means isoantigens means, what happens in isoantigens? Again, we'll take our examples only. We belong to the same species, but your blood group and my blood group is different. Your MHC, that is major histocompatible antigen, okay, you have what are called MHC cells. These are major histocompatibility. We all don't have a chapter on this, but you have to know what is MHC, which I will be telling you in the later class. So these, okay, blood group and MHC. Now see, though you and me are belonging to the same Homo sapiens, I just cannot take your blood group may be A and mine may be B. I just can't transfer your blood to myself, okay. Similarly, your my organ or your organ cannot be simply blindly be transplanted to anyone, though we belong to the same species. Why? Because our blood antigens or our histocompatibility antigens are different. So these are the example of isoantigens. Then what are heteroantigens? These are antigens belonging to two different species are called as heteroantigens. Example, antigens of plant or animal or microorganism. So you can see that we are completely different, okay. A plant or an animal is, or an microorganism do not have any much similarities with us. So when such type of antigens are called as heteroantigen.